We will be talking about how mutations can be used to our advantage with the different biotechnologies. But before we do that, I think we should do a little bit of a review for amino acids. So like Mr. Fath was saying before, there is a process that in how we end up with amino acids, right? So we go from DNA, then we move to RNA, and then we move to the proteins, but those proteins are made up of these individual amino acids. So for example, I think you should pull out your codon chart from your notes. Should have it handy. So the example here, CGA, you can find it on your chart. You can see it's related to arginine. It would be the amino acid. Next slide. Okay. So how we read our chart, just to remind you, the first nucleotide is going to be, you look along this column here. So for arginine, we had the first one there. So that was with C, right? And then we look to the next nucleotide, which was all the way over here is G. And then you notice there's a couple different options here for what the third nucleotide could be in order to give you the arginine as the product, the amino acid from that mRNA codon. So just to review, if you go to the next slide then, maybe you can try, can you decode what the amino acid sequence would be here? Maybe I'll give you a second if you can read that out. And actually we have it up on the board here, so I'll write it up as we decode it. So what do we have here? You can shout them out. What's GUU?
Valine.
Valine, okay. So I'm just going to write VAL for short. What's the next one? CAU, using your chart.
Histidine.
Histidine, yeah. Leucine.
Leucine. .309. Proline.
Okay, yeah, proline.
Glutamic acid.
Glutamic acid, perfect.
Two
of them. There we go. Nice work. That's just a little review for how to find the amino acid. So let's talk about then, what does it mean for it to be redundant? So if we can go back to the last slide for a sec. Thank you. So you notice how there's all sorts of different options that you could have the same amino acid for that, for those different combinations of nucleotides. That's the redundancy. So if for whatever reason you needed a higher proportion of a certain amino acid, chances are there's going to be more combinations in order to make that amino acid. That's something to keep in mind. Can you go back for a sec? So there's something special about these ones. Do you guys remember? So they don't have an amino acid for that combination. What does that mean? What's a stop mean? Yeah?
That's where you stop encoding the genes.
Right. Yeah, so that will be, if you're going along in a sequence, just like we had on the board there, go along, and then after that, if there's a stop codon, that's going to tell your machinery to stop doing the transcribing process, right? And then the start one here. So this one does code for an amino acid, the start codon. What is that a signal for? Start codon.
Start.
To start, yeah. So the transcription machinery is going to go and find that start codon, and that's where it's going to begin the process, right? For the translation into the amino acids. It's going to start at that start codon. Okay, next slide. Thank you. So continuous. I'm talking about redundant, how there's more than one sequence that will contribute to making that particular amino acid. Continuous is what it looks like. Is there any punctuation in our mRNA when it's made? Is there a period? Is there exclamation mark? Anything like that? No. So on the sequence that we have written on the board here, you know what? You notice I wrote those little dashes. Those little dashes don't actually exist. That's just for our notation to make it easier for us to keep track of, right? So usually it'd just be written G, U, U, C, A, U, 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 G, right? And it's not like it has a letter on it. That's just how we interpret that as people, right? That's the code that we've given to that. So it's continuous. The only punctuation we get is that stop codon, that start codon. So those are very important, right? And universal. So the proteins that you get as a result of that mRNA in your eyeball, is that going to be the same type of amino acids that are in your fingernails? What do you think?
Yes.
Yes, right? So the code, it's universal. It's going to be consistent between our fingernails and our eyeballs, or even bacteria, right? That code is going to create those proteins. So that has some pretty neat implications for when we get into biotechnologies later, like we were talking about a little bit with the vectors and bacteria. Okay, thank you. So what happens though when during the replication process, if DNA isn't copied properly? Is that going to have an effect on the proteins that result? If there's a change in the DNA itself, because it goes DNA, RNA to proteins, is there going to be an effect?
It's not gonna work.
It's not going to work? Well, yes and no. We'll find out. So there's a couple different types of mutations, like we were talking about before. A change to the DNA sequence is called a mutation, right? So there's different scenarios of whether it'll make a difference or not. So to investigate that, I'm going to hand out a worksheet here, and it's all about genetic mutations. So I'll hand you one here. I'll hand you one here. You'll see at the top, there's a list of the different type of mutations that we are going to focus on. So there could be a deletion where one of the bases is lost from that DNA sequence. There could be an insertion where a nucleotide is added, right? These are all errors from the replication machinery of DNA. Or it could be a substitution. Maybe by accident, instead of a C, a G is thrown in there or something, right? And then there's different results of that. So it could be a missense mutation where there's a change in the amino acid that results. There could be a silent mutation where it doesn't change at all and it's the same because of that redundancy, right? Or it could be a stop amino acid a stop codon is there instead of amino acid. That becomes a nonsense mutation, right? So what I'm going to get you to do is read through the worksheet, circle the mutation that is in each example there, and I'll ask you for those in a minute, and why that's important. Think if there is going to be a result that's going to change the protein out of that. Okay, so I'll give you a second. There's only 3 examples there, so in each one, just circle the mutated DNA nucleotide, and then we'll talk about why those are important in a minute. Is it clear?
So how do you find the mutation?
So you need to compare the original DNA sequence that's in the top of the box.
Oh, okay.
Oh, sorry, yeah. So please compare the original DNA sequence that's at the top in the box to the mutated DNA sequence that is given in the different examples below. You're just circling the nucleotide that makes a difference. Okay. Does that make more sense now? Sort of. It's all great. Do you have those done, or do you need another minute? One more minute. Okay, so the first one was a substitution. So that means one of the nucleotides getting substituted for another nucleotide, and it's a silent mutation. Where was the mutation in this one? Did you find it? What amino acid was affected there? So it was the third amino acid, right, in that sequence there. And it's actually listed for you on the bottom there. There's a nice little key telling you what the changes were from up above, if that makes it easier to see that. So it was the asparagine that was affected in that case, right? We substituted that A nucleotide from a G nucleotide. And as we noticed, that didn't change the amino acid. So that's a silent mutation when the amino acid doesn't actually change. Okay, so pretty nice. Is there going to be effect then for the protein that's made? No, right? Because it's the same amino acid that resulted from there. So that's pretty good. So the second example there, the substitution reaction there caused there to be a T instead of a C, right, in the second amino acid, in the tryptophan. And what did that do?
Stop DNA sequencing?
It stopped it. So is that going to have an effect on the protein?
Yes.
Yes, absolutely. Lots of the amino acids aren't there, and so the function will definitely be changed, right? And then the last example here. What was the changed amino acid?
Tryptophan changed to C.
Right. And is that going to have an effect on the protein?
Yes.
Possibly, right? Most likely, yes. It does say, yes, there will be effects. And there will be, we will be creating a different protein because of that. But if the function of the protein will change will really depend on the properties. And that's when you get into biochemistry and what these different proteins look like, how they fold, what the interaction is like. But most likely there's going to be effects. Okay, now I would like to give you guys a chance to try with this example that's on the board behind you. I'm going to assign a different type of mutation, and if you can come up quickly to decide how would you change that sequence in order to produce a certain type of mutation. So David, can I get you to do a silent mutation?
Okay.
Okay, from the sequence on the board here, so silent one. And Taylor, I'm going to get you to do a missense mutation.
Okay. I
Okay.
don't have a sheet.
Yeah, sorry, it's just from this one up here.
Okay.
Oh, you need a chart.
I do.
Thank you. And then we'll get you to do a nonsense mutation. Okay. All right. So I'll just give you a minute and let me know when you're ready to go. You don't have to write it up yet. I'm going to go first. Should I move the camera over?
Are you over here now?
Yeah.
You'll have to change your
My
slides.
slides. Yeah, that's it.
You're at 14 minutes, just so you know.
Okay, so I'll go first if that's okay. All right. So the first one we're gonna do. Are you guys ready to go? I think sounds good. Just because it wasn't on your list here, so I just wanted to show what would happen if we were going to do an insertion mutation. So that's where you add an amino acid. So I'm going to insert right here, and I'm going to put a G in there. And so are these two amino acids, are they going to be affected at all?
No.
No, but everything after is going to be affected, right? Because it's all going to shift over. So we have GUU and then GAC, right? It just continues over UCC, etc., right? So is this amino acid here, GUU, the same as UUG?
No.
Definitely not, right? So that we're changing into a valine. And then every single other amino acid is going to be changed as well. So this is an insertion mutation, and this is what we call a frame shift, okay? So it's taken our original frame and it's shifted every single amino acid that resulted after that. What about if I did a deletion? Would that make a difference? If I, instead of this here, instead of adding a G, I'm just going to take out a U. Now suddenly all of these are going to be changed, right? Every single amino acid. So deletion is also going to be a frame shift. The only exception to this is if I take out the entire codon. That makes sense? If I take out a set of 3. Or if I add a set of 3. And it doesn't even have to be that particular codon. I can take, these 3 here, and then this one would be affected, this would be affected after that. There wouldn't be effects. So. Okay, who would like to go next? Let's see what we have up here. Let's do the silent mutation. Is that yours? Which one would you like to change? Would you like to come up here to do it, or
So
just go where you are?
silent mutation is when you.
Oh, sorry, I should have put this back. That was a U, right? UUG? Oh, yeah. Okay. Sorry, go ahead.
So one of them is taken out.
So a silent mutation is a substitution, right?
Okay.
So it's a substitution point mutation that has no effect on the cell's metabolism. So this usually results because of the redundancy, right? So this is where you want to change one amino acid
something
or
else.
one nucleotide to something else, but it's going to stay the same amino acid. So one example here, if we changed the ACU to ACC, you would still result in the threonine amino acid.
Mhm.
Okay? So that's how that one works. How about nonsense mutation?
If you change any of them to like UGA, so let's say change leucine to UGA.
Okay. This one here. Yeah. UGA, and then what happens?
And then it stops coding.
And then it's a stop. And how's that going to change the amino acids that result, or the protein that results?
It would just be super short. You wouldn't get most of the rest of the information.
Right, so we're missing a lot of the information, right? So nonsense mutation is a substitution. So again, we're switching just one thing out. Point mutation that changes the codon from an amino acid to a stop codon, which actually doesn't code for anything, right? It's a stop. It's a signal more so than an amino acid. Okay, well, what did we change that from? That was UUG. And then last but not least, we have our missense mutation.
So if you changed the CAU to CGU,
Okay,
it would change it to an arginine.
U goes to arginine. And is that going to have an effect on the overall protein?
Potentially.
Most likely, right? Okay, good. And I'll just draw your attention onto the slide here, why frame shift mutation is important. You'll notice here I put a little sentence here. It says, the fat cat ate the rat. But when you take out one of the letters and you shift over the entire sentence, that doesn't make any sense anymore, does it? Right? So that has a big effect. So these mutations can happen naturally. And a lot of these mutations will actually result in different phenotypes in people, right? So if you change what the proteins that make up a person, then that's going to change how we look and potentially could lead to different disorders that we see, right? Just by a simple change in DNA sequence. This can be used also to our advantage to change and to create little mutations, possibly by fixing mutations that were naturally there in the DNA. So someone, if they have an inherited genetic disease, it's possible you can actually fix that using some different genetic technologies. So one of them I'd like to talk to you about is called CRISPR. You can follow along in the slides up top here. You don't have to worry so much about what the name means. Clustered regularly interspaced short palindromic repeats. What's most important about that is that it's a repeated DNA sequence. So this is actually part of the bacteria immune system. The repeats actually in the sequence DNA of the bacteria was the same as viruses that liked to infect it. Isn't that amazing? So they noticed these sequences of DNA and they actually thought they were kind of junk. They thought these were extra things in the DNA, but it turns out it matched the virus, which is pretty neat. So what happens is CRISPR works with Cas proteins, so CRISPR-associated proteins, and CRISPR tells Cas where to slice DNA. And that's really handy if you have an invading virus that's coming in, and CRISPR is saying, all right, Cas, which is an enzyme, right? It's telling Cas to cut at a certain point, and if a virus is there, it can cut up all the virus DNA, which is really good for its defense system, right? But we could potentially use this to our advantage. So a particular Cas protein, Cas9, it comes from Streptococcus pyogenes. Anyone know what that disease,
Staph infection?
Strep throat, actually, is associated with that one. Yeah, that type of bacteria. So it SNPs DNA. Whoops. Well, that's no fun. Excuse me. Where are we? Here we are. Yeah, so it SNPs the DNA. And the neat thing about this is if you tell Cas9 which sequence to cut, you can target a specific spot in the DNA. And how you tell it what to cut is by using complementary DNA. So I'll just show you a quick infographic here. So that's Cas9. Sorry, it's small. Computer glitch here. So that's the target sequence on the DNA in yellow. So you're going to create a guide RNA, which is complementary to that DNA. And that's going to have those repeated sequences on it, which is going to be a cue to Cas9 to come in and make a cut in the DNA. So Cas9 comes in, notices that sequence that guide RNA has bound to, and it comes to that right spot. It's very specific, right? Because you can make it up to 20 base pairs long. So Cas9 cuts that cuts that DNA strand, and it cuts it on both. And then what's going to happen is the DNA repair mechanisms that are already naturally in the cell are going to come along and try and fix whatever the problem was. So you can try and use this to your advantage, and there's actually lots of different biotechnologies that are trying to do this right now, especially CRISPR-Cas9. So you can use this with diseases like potentially with cancers, hepatitis B, these genetically linked disorders, especially those with only one mutated gene, like Tay-Sachs disease, there's only one mutated gene. So there's lots of different uses. We're going to talk about the implications of using this type of biotechnology in a town hall meeting.